Well, good afternoon, everyone. It is a pleasure for me to introduce you today, Ali Reza Balizadeh. Some of you, very few of you might know him because he visited us already in 2018. He was here for 2 months. And Ali is a physicist working in aspect of computation and neuroscience. He is professor at the at the physics department at the Institute for Advanced Studies in Basic Science, and is also researcher at the Passaregat Institute for Advanced Innovation and Solution. He says that in Iran, everything is advanced, so every title has the advance on it. He works, as I said, in computation and neuroscience. He has many international collaborations. To highlight, beside with us, he has a very nice collaboration with Peter Tass from Stanford University in the medical department. So the aspect of computation and neuroscience he works on are very interested also from the clinical point of view. So Ali, thank you very much for being here, and the floor is yours.
Yes. I don't have a pointer, so maybe thank you very much. So thank you for your kind introduction, and thank you for this opportunity that you gave to me to present my work here. Indeed, first of all, I should say that
Is the microphone on? The microphone is...
Yes, it is. Yeah. Indeed, I changed the title of my talk a bit. What I sent to you, and it is in the flyer, is information processing in oscillating brain, but I changed it to processing by oscillating brain to insist on the role of the oscillations in the information processing in the brain. We don't say that oscillations are there and information is processed in an oscillating media, but we say that oscillating is important in defining the ways that the brain processes the information. Maybe all of you know that elements of processing information in the brain are neurons, neural cells. The speed to very large complexity in the dynamics of the neurons, we can have a simple look on them as excitable system. Excitable system means that if you excite these elements, the neurons, with a small input, it doesn't show any response or small response. But if the input exceeds a threshold, you see an upstroke. It doesn't work yet. Should I? Here. Thank you. Great. Let me first say that the information in the brain is basically coded by the voltage across the membrane of the neurons, the voltage that is between inside and outside of a neuron. When you excite the neuron with an input above a threshold, there you see an upstroke in the voltage, and we call it an action potential, and we say that the neuron is activated or it has produced a firing. We can roughly say that the waveform of the firing in the neurons, at least for the similar neurons from the similar type, of the same type, are almost less and more similar. So the information is not coded in the waveform. Instead, what is important is the timing of these events, which is called action potential, and lasts for roughly saying about 1 millisecond. A conventional way of thinking about the how the neurons code information with the timing of spikes of the events is that they change the rate of the firing, the rate of the activity, often the change in some features in the stimulus. For example, this classical experiments of Hubel and Wiesel showed that when you change the orientation of the lines in the visual field, there are some neurons in your primary visual area back of your brain that change the firing rates depending on the orientation of the lines in your visual field. When the lines are in a specific, which is specific orientation, the neurons show maximum firing rate. They are maximally activated. And this can be quantified by a curve that is called tuning curve, which is very important for the coding in the brain. And in the horizontal, you have a feature in the stimuli here, this orientation of the lines in the visual field, and here is the firing rate. And many of the neurons in the brain respond to a definite feature in the stimuli. It can be the color of the objects in the visual field, the orientation of the line. It can be the frequency of the sounds that you hear, and many other features. I should say some other facts about the brain. The first fact is that the structure of the brain is modular, which means that you know that what we mean from the modular network. The modular network are sort of the network that are divided to some modules, some subpopulations, that inside the population you have dense connection and outside the connections are sparser. Other ways of definitions are there, because when we say that the brain is modular, we mean that not only we have connectivity, a modular connectivity in the brain, but there are other differences between different parts of the brain, from the connectivity inside and outside the modules, type of the neurons and properties of the neurons. They have different chemical agents in different parts of the brain, and also the genes that are expressed in different modules are different. So different properties define the modularity of the brain. Not only the structure of the brain is modular, but this brings up sort of functional specialty. It means that each part of the brain, roughly speaking, does a definite function. For example, here in the back of your brain, you have areas that decode and encode the visual, the visual inputs. Here in the side of your brain, we have auditory area. On top of your brain, you have the areas that neurons that encode the somatosensory inputs. Here in the front, as you may know, you have association network, association region that sum up the inputs from other brain regions and is involved in higher brain function like decision making, attention, conscious perception, and so on. What is important is that, roughly speaking, again, each part of the brain connected to the modular structure of the brain has a different function. Another consequence of the modular structure of the brain is that in microscopic level, the neurons that are close together do the similar function. They receive input from the shared sources. They have a very strong connectivity between themselves. and they have similar tuning curves. Here is a demonstration of the tuning curves of the neurons in primary visual area back of, as I told you. And you see that the colors show the orientation that they respond maximally, and you see that the neurons are nearby, have similar tuning curve, meaning that they respond to the same orientation in the visual field. And this is true for other regions of the brain, meaning that neurons that are nearby are strongly connected, receive input from many shared sources, and also do the same function. The tuning curve is less and more similar. Another fact which is worth noting about the brain is what you usually hear, that the information processing in the brain is parallel. It means that when an input, let me say that we have a region in the brain, in the forebrain, beneath the cortex, the outer part of the brain, which is called thalamus. You have heard about it. The conventional role of the thalamus is to receive inputs from the periphery, from the sensory areas, and distributes it in the relevant areas in your cortex. After information comes to relevant part of thalamus, it is distributed to the cortex. For example, here I've shown the visual circuitry, the system that codes the visual input. And you see that there are two main actually pathway. One of them, which is what pathway, encodes the object, the color of the object, the lines, the shape of the object, and one pathway is dedicated to the which is called the where pathway, and decodes actually the position of the object in the visual field. And these are segregated in the beginning of the information processing of visual inputs in the brain. Another fact that should be mentioned is that not only the information processing is parallel in this way, this is also hierarchical. Here, with hierarchical, I mean a serial information processing. You see here that after the information comes to the primary visual area, primary sensory areas, that are those areas that receive directly input from the thalamus. They receive directly the input from the sensory system. After that, there are pathways that each layer, each region in this pathway decodes some different properties, different features of the input. Usually, how much you go further deep in this processing hierarchy, different features, actually more complicated features are coded. For example, here, the first coding level decodes the lines, the orientation of the lines, and in the next level, the shapes are coded, objects and faces in this visual. You see that in each level of computation of the information processing, the features that are processed by that region, by the specific region, becomes more complicated. Maybe say some terminologies about which I use today. There are also examples about other sensory modalities, in other sensory modalities like auditory modalities, somatosensory, and olfaction. You also see the hierarchical, it means that serial levels of processing. So that is a general feature of the processing in the brain that are both parallel and are hierarchical. Another fact is that how much you come from the back. This is the rule of thumb. How much you come from back side of your brain to the forward side. The level of processing becomes more complicated. Simple features in the backside of your brain and how much you come to the forebrain, more complex features are encoded. And at the final region here, in the actually prefrontal or frontal part of your brain, the function of this part is that it integrates the information from different modalities, from from visual information, from auditory information and other information, and associates them and makes high-level decisions. It controls actions, controls your, in some parts, your emotions, your decisions, and so on. But what is a consequence of this connectivity? As I told you, we have dense connectivity between the nearby neurons in the brain. This dense connectivity and strong connectivity, we know that brings a correlation between activity of the neurons. So the first consequence of this type of connectivity, modular connectivity, which means that we have in the microscopic level, we have a very dense connectivity, is that the neurons activity there is correlated. And correlated activity of the neurons, I have shown each activity, each action potential here. We have here 100 neurons, and each activity is shown by a dot. The result of this correlated and synchronized activity is the appearance of the oscillations in high amplitude oscillation in the network level. This is what we roughly measure in the experiment with EEG signal or LFT signal, whatever. If you put electrodes outside or inside the brain, what is measured by this electrode is the mean field or network level activity of the neurons. And if you see some oscillation, high amplitude, large amplitude oscillation there, it is naturally a consequence of synchronized and correlated activity of the neurons. For the years, from the middle of the previous century, people has identified different type of oscillatory behavior in the network level. They have characterized them in different ways, and we have now they have observed different oscillations with different properties and mainly with different frequency, from very low frequency oscillations from sub-hertz to high frequency oscillations, which are roughly higher than 30 hertz or higher than. And these two gamma oscillations, that theta oscillation, it's in the range of 4 to 8 and higher than 25 or say 30 hertz are important in the examples that I gave here today. The point that should be noted is that, as you may guess, this structure, modular structure, and its consequence, which is synchrony, reduces the information content of the brain. It's obvious because when many neurons do the same thing, the information that can be processed in the brain reduces too much. But what is important, as we will talk more today, is that this type of activity facilitates and enhances the communication between different parts of the brain.
Excuse me, in the previous slide, the statement says something which is experimentally confirmed, just a theory hypothesis for what is.
Which one?
Synchrony reduced the information capacity of brain networks, but it facilitates indication.
It's theoretically, many people have studied that, and
people believe that this.
Yes, yes, know that.
It's a hypothesis.
more than a hypothesis, because you know, the dynamical repertoire that a system without synchrony can produce, it's very much larger than the system that is synchronous.
It's not confirmed in experiment, it's just a hypothesis, not a field.
Maybe we can talk at the end of, we can talk about it at the end of my. But again, I insist that that is more than a hypothesis.
Sorry, what does communication mean?
With communication, I mean that information sharing or information transfer between regions of the brain. The different parts of the brain talk with each other and transfer information between, there are transfer information between different parts of the brain. And with communication, I mean this sort of information transfer. To say just something interesting about the role of the oscillations in higher level functions of the brain, I just want to say something about the working memory. Working memory is a type of short-term memory that you have experienced. It may be if you read some digits, some numbers from one page and you keep it in mind and type it in another page, a telephone number or a code, you keep it in your working memory. The speed to the long-term memory that is believed to be saved in the strengths of connection of the synapses in the brain, this type of memory doesn't change any structural property in the brain, and it only is maintained in the sustained activity of the neurons. You may know that after classical experiment of Sternberg, if you have heard about it, the digits that can be stored in your working memory is roughly 7 or 6 or 8 and like that. And considering that, considering that with the high and low frequency components that I talked about, you see in different brain areas simultaneously gamma oscillations over the 30 hertz and theta oscillation between 4 and 8 hertz. And the number of cycles that can occur, the number of high frequency cycles that can occur in every single low frequency oscillations is about 7. And people believe that this is a hypothesis, is not proven yet, but we have some elementary evidences for that, is that each cycle in high frequency oscillations maintain one of the digits in your working memory. And that the limit for working memory is that this number of high frequency cycles in low frequency oscillations is limited to 7 or 8 or like that. Another important experimental result is that the information which is transferred between brain areas as measured by experiment are dependent to the frequency. One important example is about, again, in a working memory task, which shows that we know that in forward transfer of information, with forward I mean from the low primary areas of sensory system to high areas of the system. This is the forward flow of information, and we have backward information flow from the frontal part to the primary areas. It is known that, and we know that your cortex has several layers, 6 layers roughly speaking, and the superficial layers are responsible for the information flow in forward direction, and deep layers convey information in the reverse, in the backward direction. And it is shown that in the forward direction that is characterized by the superficial layers, the amplitude of high frequency gamma oscillations is larger, where in the deep layer, the amplitude of lower frequencies are larger. This has been seen in different tasks in different species. For a visual task, it has been shown as by other group that again in superficial layer you have a maximum power in the high frequency range, and in deep layer, which is responsible for backward flow of the information, you have maximum power in low frequencies. And more of that when the people measure the information transfer based on experimental time courses of the mean field activity, they have shown that information transfer is backward level, is mainly conveyed by high frequency component of the activity, where the backward flow of information is conveyed by the low frequency component. That shows that having different oscillatory components in the activity of the brain provides a way for the brain to send information in different direction over different frequency range. Let me just say another, introduce you another region of the brain, which is hippocampus. Maybe you know hippocampus is mostly famous for its role in spatial navigation. Maybe you have heard that the London taxi drivers, and formerly, not now because we have now GPS, but it was an experiment that showed that hippocampus of the taxi drivers supposedly in many large cities are larger than other people because its role in the spatial navigation. In the hippocampus, based on experiments on the rodents, we have some neurons that code the location of the animal in a field which is known for the animal. If this rodent is here, some neurons fire there, fire with higher frequency, and if the rodent changes its location, different neurons increase the firing rate. But this is classical view, which information, there is a map between the location of the rodent in the field, in a known field, and the neurons that are firing. But as I want to say that this is the classical view, because beyond the firing rates, what we see in the activity of hippocampus, these regions of the brain, is the presence, again, multiple frequency oscillations. We have a low frequency, again, in theta range, and high frequency in the gamma range. And what is seen here is that in some definite range of the phases of low frequency, the amplitude of high frequency components are larger. Which means that there is an interrelation between the amplitude of high frequency oscillation and the phase of low frequency oscillation. This is not only the case for hippocampus. In other, here is again hippocampus, but in other regions of the brain, including cortex of rodents, olfactory system, olfactory areas, and also in EEG of the human brain, we see this type of activity, a low frequency activity that modulates the amplitude, the higher frequency activity. What is the consequence of this type of activity in the coding? With having low frequency activity, as people say, and have some experimental evidences, provides a time reference for the activity of the neurons. So here, the time of the activity, of the firing of the neurons has another meaning beyond the firing rates. Regardless of the firing rate of the neuron, which is high or low, the time of firing of each neuron related to the phase of the low frequency oscillations conveys information and indeed has information about the location of the rodent there. It is worth to say something also about the pattern of oscillatory activity in the healthy, in the normal brain, and in different diseases. I have just brought here two examples. One is that this is, you supposedly you understand that this is a phase. This is a task which is called the Mooney phase, after Mooney, a psychologist that designed the task. It is very difficult to understand that as a phase. Many brain areas are involved to understand that this is a phase. And it is shown that after exposure of the brain to this picture, some oscillations in different parts of the brain in the gamma range appear 200 milliseconds after exposure of the brain to this stimulus. It is seen that the people with schizophrenia cannot understand that this is the phase. And it is shown that this high frequency activity is much reduced in these people. And another interesting result is that, do you understand? Can you say what is this? Yes, it is yes, upside down. But I cannot understand it. If you can understand that is again a phase, these oscillations appear like this. These oscillations are correlated with your understanding that this is a phase or not. So appearance of oscillations in some part of your brain depends on if you have understood this, perceived that as a phase or not. And another interesting or another disorder is that we know that in people with Alzheimer's disease, the power of the gamma oscillations in some tasks, in some states of the brain decreases. In the model of rodents, which are genetically become a model for Alzheimer, they have shown that this is the case again for these rodents. The amplitude of gamma oscillations compared to wild type healthy ones decreases. These people showed that if you excite the brains of these rodents with an oscillations excitation in the range of gamma oscillation 40 hertz, you can change the time course of the disease. It means that we know that, maybe some of them, you know that we have a toxic agent, beta amyloid in the brain, which when it increases in the brain, it causes the death of the neurons, and it begins, it is a cause, it is believed, it is a hypothesis that this is the cause of Alzheimer's disease. These people show that if you excite the brain with a 40 hertz oscillatory input, the concentration of beta amyloid in your brain decreases, not in your brain, in the brain of this mouse models of the Alzheimer. Here is the concentration of beta amyloid before excitation with 40 hertz. Here is the concentration of beta amyloid after. And if you change the frequency of oscillation, an increase in the amyloid beta is seen. This means that it is very important study because it shows that if you excite the brain with an oscillatory component, which is lost in the disease, you can change the course of the disease. Now, we have very good documents, very extensive documents about what happens for the oscillatory patterns of the brain activity in different diseases, different neurological and cognitive diseases, which is used sometimes by the physicians to identify the diseases. Sorry. Just 10 minutes?
Yeah, 10 to 20, 10 to 15, 20.
Okay. So now I think that we are the long introduction has finished, and I can say what is the hypothetical role of the oscillations in the brain. Maybe you are familiar with this picture. Engelbert Kampfer is who for the first time, he was a physician, Dutch physician, who traveled to East Southern Asia and reported that there are trees there that on the trees you have some fireflies that flash together in synchrony, and you can see the light of these fireflies from the kilometers away. Also, David Attenborough has a documentary about it in BBC documentaries. And another example I want to bring up here is the voice of the clapping of the people in the concert hall. These people has shown that after the piece of music have finished, people have began to clapping, and what is received by a microphone there in the concert hall shows some high amplitude oscillations after a while, after 2 seconds. That means the synchrony of the clapping that is a common experience for all of you. And first, it is interesting that with just a microphone in a concert hall, you could publish a Nature paper. I don't know about the details, but it's interesting. But in these 2 examples, you see that synchrony in nature and by human is used to increase and amplify the signal with when the fireflies flash in synchrony or people clap in synchrony, they provide more strong signals that can be more influential for other people or other animals. This is also the case, it seems that this is also the case for the brain. A fact in the brain is that the coupling between every 2 neurons is so small that it is not possible that firing an activity of one neuron can exceed activity in other neurons. We need many neurons to spike in synchrony to excite a target neuron. This is what that led to a concept that is seeing fire chain. This is a layered network, exclusively feed forward. The connections are feed forward, and in each layer we have a number of neurons, and it is shown that if the neurons in each layer fire in synchrony, that then the signal can be transmitted in this hierarchy. But if they are not synchronized, or the number of neurons that are firing synchrony are small, the signal is faded and cannot be transmitted. It is interesting that there are some experimental results that can be related to this. This is for locust olfactory bulb, olfactory system. It's shown that after exposure of the animal to the olfactory inputs, some oscillations appear in that part of its nervous system. And also in other animals, in the cat, the very classical experiment is showed that exposure of the cat to the or visual stimuli elicits some oscillations in the gamma band in its visual system. It seems that these oscillations are a consequence of the synchrony of the neurons that are involved in the transfer of information in the hierarchy that I showed you in the first of my talk. And they help to the reliable transmission of the signal in this hierarchy. What is the consequence of this phenomenon in higher level brain function? In many sensory tasks, we have some, we should choose which part of the stimuli should be chosen to be attended. For example, I have here, I want to attend to that picture, to face a view, and there are competing inputs that come to the brain, and just a few of them are chosen by the brain to be processed by a conscious process. These people showed that the speed that these are, these are tuning curves. We have 4 populations with different tuning curves, and this part should be attended, say somewhere in your visual field. And they showed that even if the mean firing rate of the neurons in 4 channels are the same, this channel, because of the firing, are more synchronized, is the winner of the competition. And this channel can be attended while other channels are ignored. So far, in the previous slides, I showed you the same fire chain, an exclusively feed-forward network that synchrony led to the transmission of information there. But we know that the connections in the brain are not feed-forward. It's interesting that almost 80% of the connections that each neuron receives is from the nearby neurons in the same layer, not from the forward connection, not from the backward feedback connection. This is interesting. We call them recurrent connection. This recurrent connections actually define the state of that population that receives input from outside or from higher level areas. It is shown that for almost realistic models of biological neural network, that presence of these recurrent connections gives rise to some damped oscillations following the exposure of the system to a pulse packet of inputs. This damped oscillation provides a new way to transmit information. If this is the first pulse packet that cannot be transmitted, here is each layer of the network in a hierarchical level are like this, and this cannot be transmitted, as you see. But if the next pulse packet is received by the first layer in this window of time, which is again the network is highly excitable, the signal has more chance to be transmitted. This was shown for the first time by Han and et al., which actually unraveled the role of the recurrent connections, not in the direction of the flow of information. As an extension of this work, we showed that we questioned how this train of pulse packets are produced, and we hypothesized that presence of feedback connections between the first 2 layers of the feed-forward network can make this input signal to reverberate between these 2 first layers, and then an oscillation is seen in the activity of this network, and the propagation can be successful, provided that the delay of transmission of the signal between these 2 layers is compatible with the period of these damped oscillations, which this is an external property of the connection, and this is a property that is defined by the recurrent connection inside each population. And I ask you to compare these 2 pictures from experiment and what is seen in the networks with feedback connections with right delays. So what can I say for now is that obviously synchronous activity has more chance to be propagated, and neural oscillations can facilitate, and I would say that they can also control the transmission of the signal and information exchange between different populations. And as I showed that feed-forward, recurrent, and feedback connection can have different role in transmission information in the forward direction in the brain. 5 minutes I have, or? Yeah. Good. So I can. complete mind. But before I proceed, I want to talk about another concept, that is structural, functional, and effective connectivity of the brain. We talked about structural connectivity, which is modular in different spatial scales in the brain. That's the presence of physical connection between every two brains or between two regions of the brain in different spatial scales. But functional connectivity is also defined, as you call it, maybe I saw in a poster in the institute, you call it correlation matrix. That is the similarity, statistical similarities between the activity of two parts, two neurons, or two regions of the brain. If we measure the correlation between activity of two parts, and we can then define a functional link that is quantified by the amount of correlation between the activity of two parts. This can give rise to functional connectivity, which is different, can be different from structural connectivity. A type of functional connectivity which is defined upon the activity of the different part of the brain is the effective connectivity. The functional connectivity is based on correlations, so it's not directional. It has no direction and doesn't show any causal effect. But effective connectivity is based on the causal effect of the activity between any two parts of the brain, and it defines a directed graph. So we have three different types of the connectivity: structural connectivity, which is physical connectivity, and two types of functional connectivity, which are defined based on the similarity of activity or causal effects in the activity of the neurons in different parts of the brain. It is shown that while the structural connections are less and more fixed, at least in the short time scales, the functional and effective connectivity are subject of the change over time. It is shown that if we have in the resting state, when you don't do any cognitive task, if you construct a functional connectivity like this, if you change the task by attention state or another task, memory state, the functional connectivity in your brain and effective connectivity, of course, changes. So there are also other examples. Many subnetworks are defined based on the connectivity between other parts of the brain that are functional, based on the correlation or information transfer between different parts of the brain. Another interesting result is that not only functional and effective connectivity are time-dependent, state-dependent, the speed to the fixed structure. They are also frequency dependent. In this experiment based on EEG signals, people, and I'm sorry, they have shown the information transfer. Warm, hot colors show the source of information in the data, and the blue colors show the target areas. They have shown that while in low frequency range, that, for example, here is the source of the information. In the high frequency range, right, like beta, the source of the information has changed. It means that effective connectivity here, because it is directed and based on information transfer and causal effects, change is dependent on the frequency. But how? How the model can help to explain this experimental result. There is a very fascinating and influential theory about the role of oscillation in control of the connectivity, functional and effective connectivity between different parts of the brain. If we have two oscillating regions, and if they are in a good phase relation with each other, when one part is active, the other part has open eyes, which means that its excitability is high and it can successfully receive the information from other parts of the brain. But if they are in a bad phase relationship, when this is active, the first part is active, the eyes of the target population is closed. So while there are structural connections here and here between every two pairs of population here, you can switch off the channel for information transfer by changing the phase relation between two parts of the brain. And we had actually some simulation that in extension of this theory and hypothesis with both of them is with Claudio. We showed that with the fixed structure here, we considered the structural connectivity of the different brain regions based on the MRI data and used it as a network, and we put some population on each network that represent the neural network in that region, and we showed that if the each population that has a higher frequency, slightly higher frequency compared to other parts, can take the role of the source of information. So it's possible that changing the frequency of oscillating activity of different parts, you can change the sources and target, you can change the direction of the information transfer in a dynamic way. This is also shown by other people in another modeling and theoretical experiment. What was interesting that in continuation of that work, we considered again delay in the transmission of the signal, which are inevitable in the brain. I can say that because of almost small speed of the information transfer of the signal transmission between two parts of the brain, there are actually delays in the transmission of the signal which are less than 10 milliseconds, which is not small, considering that the processing in the brain, the time scale of processing in the brain is milliseconds. So 10 milliseconds and in this order of magnitude is not small and cannot be ignored. We questioned how this delay, when we change this delay in the interaction between two populations, changes the direction and the amount of information which is shared or transmitted in these two. Here we have delay of the connection, and here we have, like previous study, the difference in the frequency of the oscillations of two populations, and we showed that depending on delay, for very small delay, that was the previous results, is through the higher frequency regions send information to low frequency regions. But the increasing delay, we identified some regions that speak to the presence of the difference between the frequencies, the information transfer is symmetrical. And also there are some regions of some range of the delay that the information flows from low frequency to high frequency. But what is important, and we showed it, actually we proved it with some mathematics. And what is important to say that delays are structural properties and are fixed. So what is the meaning of changing delay here? Indeed, delay is not, absolute value of delay is not important. What is important is the delay compared to the period of oscillations. So when the delay is constant, if you change the period of oscillation, the frequency of oscillation, say in low frequency range, in high frequency range, the effect of delay changes. And this is the way that we've shown that we can have different preferred directions of the information transfer in different frequency ranges. So let me to skip this one because I'm out of time. So, and to conclude, maybe I should say something that indeed we don't know what is the exact mechanism of processing information in the brain. This is not what I'm saying. I'm not justified to say that, but it is told by the pioneers of the field. The brain sciences and these theoretical views to brain sciences are very infant. So we still don't know what happens there. So there are many hypotheses, speculations, and there are plenty of experimental results that in some cases they're contradicting. But I can say that from a black box that brain used to be in the beginning of the 20th century, now it is turning to not a transparent box, but to a almost transparent. But still, as I say, all the theories that are posited by different researchers are still far from proven. But if there is actually the mysteries of the brain, if there is a progress in unraveling the mysteries of the brain, it has a very high impact first in the changing the paradigms of AI. Next, if we know how the brain processes information, then it is more possible to have brain-computer interface that is very important in defining the future of the human life, and most of all in treating the brain disorders. So let me thank to my collaborator, Claudio, Ingo, which is, I'm afraid he couldn't attend here, Ad Ertzen, which is now an emeritus professor, that used to be in the Freiburg, and he is one of the pioneers of the field of computational neuroscience, and Arvind Kumar from KTH, and also my students, RF Paris and Hedi Erzak. Thank you all.
Thank you, Ali. Any question or comment? Yes, Annalisa? Okay. Because there are people on the line.
Thank you very much for the interesting talk. I have a curiosity on the first part. That is, what happened to the hierarchical structure if there is a damage on one of the parts? So
can you repeat?
What happened to the hierarchical structure if there is a damage of one of the parts? I mean, how the functionality change?
It's a very good question. Indeed, certainly something happens there. If there is only that is the only way that information is transferred. But you know, the people know that experiments show that brain is, in a limited extent, robust to the external to the damage. What happens there? What is important to know that these simplified pathways are not the only pathways that are there. There are many bypasses, many different, you know, we show that every population is connected to the next level, but it's not the case. Every population is connected to next level to the, and directly to the next neighbor, not. And these maybe, I can just say maybe, presence of this different pathway between two regions makes, in some extent, the brain robust to the damage. Maybe, and it's the case.
Thank you, Emilio.
I always ask myself why the alpha, beta, gamma, all those modes of motion are important. You are talking about the oscillating brain. Is this something that is really the oscillating ideas and the alpha, beta, and all this type of description comes from Fourier analysis. So people look at the electroencephalogram and look at Fourier analysis and describe this. Is this really the optimal basis to describe the brain activity, or perhaps other types of waveforms other than sinusoidal will describe better the kind of information transfer in different parts. So what is that historical, or there is some or simplicity, or there is something more deeper into the description of the oscillating brain by Fourier modes?
Actually, let me say that this type of definition, this type of, dividing the frequency range to different frequency range as gamma, and actually is not consensus on them, because sometimes you see that gamma ranges from 30 hertz, sometimes you see from 25 hertz. And what is important there, they are not only divided by the frequency. That is the function that people are, for a long time, the people are experiencing different part of the brain in different states. And what is called gamma oscillations is not only based on the frequency. The state and the region and the task that the animal is doing is important, and that's why these are not, they are not a consensus at them, but have a much more robust basis other than frequencies. But as you say, maybe another way is not to just considering them as a Fourier component. I have recently seen a paper about the gamma oscillations. Title was Beyond the Frequency, as it the people there stated that the waveform of different of the gamma oscillation in different part of the brain is informative. So people are thinking about that.
Sure, Ernesto.
So maybe back to your
No.
challenging question.
No, not returning to that. But your main conclusion of the first part is that, if I understand correctly, synchronization facilitates propagation.
Yeah.
I didn't understand propagation of what, and I didn't understand what is propagation. But if I assume that you are talking about propagation of information, and I assume that you are talking about an environment with 80% water, I could assume that this could be diffusion in high percentage. So in this particular scenario, I'm not completely surprised about this finding, because synchronizability in a network depends on the algebraic connectivity of the network. The highest algebraic connectivity, the better synchronizability of the system. Well, it depends on other parameters, but let me simplify. And the speed, the rate of convergence to the steady state of the diffusion also depends on the algebraic connectivity. The highest algebraic connectivity, the fastest goes to the diffusion. So synchronizability in a network facilitates diffusion in somehow. So let me put it another way, because what I am thinking is, do you think that a network that facilitates synchronization also facilitates the propagation of whatever is propagating over there?
Let me begin from another point of view, and that relates to your previous question. We know that it can be said in simple words that presence of synchrony reduces the amount of information that is processed by a network. People say that we have about 100 of billions neurons in the brain. The different dynamical state that can be produced by this number of neurons is astronomically huge. Modular structure and presence of synchronized activity very much reduces the dynamical repertoire of the network. It means that in the course of the evolution and in developmental course, people say that among the very high dimensional space that the brain can produce actions, it chooses very low number of this, very small, low dimensional manifold. It chooses very small dimensional manifold of the very high dimensional space that it can produce. It comes to so we have this cannot be considered as a deficit, because we don't need this huge number of actions. We just need some, small fraction of action that can be produced by the brain. This is the first one. But as I told you, and as you mentioned, it's obvious that if you have more synchrony, you have more powerful signals, and this is more likely to be propagated reliably. But if you increase the synchrony too much, which is the case for some disorders like epilepsy, like Parkinson, but the amount of information reduces more than what is needed for the brain. So there is a trade-off between propagation, communication, and the information which is coded. People say that there is a normal trade-off between these two. I don't know if I have answered your question. We can still.
At least I think it is well known that some, in the cerebellum or even in the dental gyrus, there are neurons that are almost disconnected. And those are areas in which the computation is huge. You do a lot of computation there and you have hardly connected neurons, so the connection is very, very low. So it looks like that you need some kind of synchronization, you need some kind of connection to promote synchronization, but then you don't want to all the neurons doing the same, because then you lose the capability of doing more complex tasks. It looks like, and this is known experimentally, but then the interpretation is another story, as you know. So still we need theories to better understand what's going on and to be sure that we are interpreting the experimental result correctly. Yeah?
I was just referring to the same there.
Yeah, I know what you're referring to.
So this is like a limit.
Yes. Well, I mean, that's right. We cannot demonstrate the theorem, but we have some results that show that. But for sure, there might be others that contradict that aspect as well. So I will not.
It can be questioned that what I talked about is the propagation of the signals, the activity. What is how the information is defined? Maybe that's your question. It's another question. And it can be questioned if the propagation of the signals means better propagation of the signals, of the activity means propagation of the information. I think that based on what I told you about the trade-off between information and propagation, there is somewhere between, when you change this synchrony, somewhere between then this amount of information that is propagated is maximized.
Well, any other quick question or? Yes, of course.
And information.
Oh, that's a very good question. We don't know. Yeah, we still don't know. I can say something that we have some, consider a population that is responding to the different part of a stimulus. So it is meaningful that which neurons are activated. People say that it is not the activity of the whole network, but those specific neurons that are activated. So what I talked about here is the mean field activity of the network, but details are inside there. A cycle in the, maybe I can show you here and it can help. I don't know, but it can help. Here, you have on the mean field level and the network level, we have some oscillations in high frequency level. But what people say that each oscillation has different meaning. Different neurons are activated in different cycles of oscillations. So if you can consider, hypothesize that if in all the cycles, same population are activated, the information that is decoded by the system is low. But when in each cycle, on the network level, cycles are similar, but the neurons that are activated are different. So the signal which is transferred in both cases, if all of them are the same neurons or different, what I see in this experiment, in my experiment, are the same. I just see that the signal is transmitted. If you want to go beyond the signal transmission, you should consider these details. More than that, we have some measures, information, transfer entropy, mutual information, asymmetry in the mutual information that measures the transfer of information, regardless of the signals that is transmitted. It's different than that. And we have shown here, maybe with Claudio, that the signal transmission in our case, in our model, is very similar to the information transfer. Let me show you here. Here in the, we calculated here transfer entropy between two populations, here the signal transmission. But they are less and more similar.
Okay. Well, thank you very much. Thanks again, Ali. Okay.